Hello students, welcome to EPG Pathshala program. I am Dr. Shoaib Lukman from the Council of Scientific and Industrial Research, Central Institute of Medicinal and Aromatic Plants, Lucknow. Today we are going to talk on the module Biosynthesis of Lipids, Part 5 from the paper Lipid Metabolism. The objectives of the paper include the biosynthesis understanding of the eicosanoids, how do prostanoids and leukotriene pathway operates, and what are the implications and significance of bile acid and its synthesis. Within the cell, eicosanoids are synthesized as and when required without any storage. They originate from the fatty acid that craft both nuclear as well as the cell membrane. The oxygenation of the fatty acids results in the production of eicosanoids, including the prostanoids as well as leukotrienes. And this production is supported by the action of two enzymes, the cyclooxygenases, or what we abbreviately called as COX enzymes, and lipoxygenases, or the LOX enzymes. The prostanoid group of eicosanoids are produced by COX, whereas the leukotrienes are constructed by LOX enzyme. The biosynthesis of eicosanoid begins when cell is triggered by the stimulus such as the cytokines, the growth factors, or even the mechanical trauma, followed by the discharge of phospholipase enzyme at the cell membrane, with an ultimate movement to the nuclear membrane. At the nucleus, the phospholipids and their esters is hydrolyzed by phospholipase A2 and diacylglycerol by phospholipase C, resulting in the release of essential fatty acids, which is of 20 carbon moiety. The step of hydrolysis emerges to determine the rate of eicosanoid formation. As we all know from the previous lecture that fatty acid give rise to diacylglycerol, and this diacylglycerol as well as phospholipids, with the action of phospholipase C and phospholipase A2 respectively, give rise to a very important intermediate in the eicosanoid biosynthesis, that is arachidonic acid. Arachidonic acid further, through various transformation and conversions by different enzymes, give rise to prostaglandins, and these prostaglandins further metabolize to prostacyclins as well as thromboxane, and these group of prostaglandins, prostacyclin and thromboxane contributes the prostanoid biosynthesis. Whereas on the other hand, arachidonic acid also gives rise to SPETE, which finally results in the formation of leukotrienes. The release of fatty acids happens due to a type 4 cytosolic phospholipase A2, which is a strategic player in the icosanoid biosynthesis, especially in the formation of arachidonic acid. The cytosolic phospholipase A2 is explicit for phospholipids, which contains GPLA, EPA and AA at SN2 position. Captivatingly, the cytosolic phospholipase A2 may perhaps helps in the release of lysophospholipid, which happens to become platelet activating factor. Reactive oxygen species and peroxidation also has an important role in the icosanoid biosynthesis. The fatty acid is symmetrical and the ensuing icosanoids are chiral. The high stereospecificity of free fatty acids allows the oxidation following any one of the described pathway, and molecular oxygen is being added to the icosanoid pathways via cyclooxygenase or lipoxygenase activity. The lipid peroxidation is perilous to the cells predominantly, whilst adjacent to the nucleus. And now, the intricate mechanisms are known which can avert the superfluous oxidation. The phospholipases, the cyclooxygenases, and the lipoxygenases are firmly controlled along with 8 other proteins that get activated to synchronize the production of leukotrienes. Most of these leukotrienes subsist in multiple molecular isoforms. The oxidation by lipoxygenases or reactive oxygenase species and the highly reactive peroxides as the primary product in icosanoid production. The leukotriene A4 is capable of forming adduct with the DNA of the tissues. The lipoxygenase also engenders cellular damage and association of lipoxygenase 15 isoform, which has been found in the pathogenesis of atherosclerosis in the murine models. The process of oxidation in icosanoid production is partitioned, which limits the damage caused by peroxides, carrier proteins, epoxide hydrolase, and glutathione transferases are the enzymes responsible for the icosanoid biosynthesis. The functions of these enzymes are predominantly involved with detoxification process, suggesting that icosanoid signaling might have progressed from the cellular reactive oxygen species detoxification. If you look at the structures of the icosanoids, we can see the structure of prostaglandin E1, thromboxane A2, and leukotriene B4 is quite similar. Whereas the structure of prostacyclin I2 and leukotriene E4 is peculiar. In the prostanoid biosynthesis process, the catalytic conversion of the free as well as essential fatty acids by cyclooxygenase is carried out in two-step process. In the preliminary step, two oxygen molecules are supplemented as two peroxide linkages and a 5-membered carbon ring is counterfeited in the vicinity of the fatty acid chain. resulting in the formation of an unstable, short-lived intermediary prostaglandin G or PGG. When single oxygen is shed from one of the peroxide linkages, the formation of prostaglandin H or the PGH takes place. Additionally, prostanoids like PGH1, PGH2 and PGH3 is evolved from PGH by the catalytic activity of dissimilar enzymes. Looking at the steps of the biosynthesis of prostaglandins, enzyme PGH2 synthase or cyclooxygenase or peroxidase converts the arachidonic acid to prostaglandin H2. Again, this prostaglandin H2 get transformed to prostaglandin D or PGD by the catalytic activity of PGD synthase. The PGH2, when acts on by PGA synthase, it results in the formation of prostaglandin E, and this prostaglandin E in turn form prostaglandin F. Similarly, the prostaglandin H2 forms prostacyclin PGI2 with the help of enzyme prostacyclin synthase. Finally, prostaglandin H2 give rise to thromboxanes TXA2 by the catalytic action of the thromboxane synthase enzyme. Now the prostaglandin E, prostacyclin PGI2 and thromboxane TXA2 are the three classes of the icosanoid biosynthesis with idiosyncratic rings. 5-carbon ring prostaglandin H are being linked to 2 oxygen peroxide. In prostacyclins, the 5-carbon ring is adjacent to another ring with oxygen, while in thromboxane, a 6 ring is linked to a solitary oxygen. The cyclooxygenase enzymes, arachidonic acid, a polyunsaturated fatty acid, gets converted to prostaglandin H2, which is a precursor of prostanoid, by the cyclooxygenase enzymes. The cyclooxygenase enzymes encloses 2 active sites, a cyclooxygenase site where prostaglandin G2 is formed, and a heme site with peroxidase activity, which is accountable for the diminution of prostaglandin G2 to prostaglandin H2. A hydrogen atom is abstracted from the arachidonic acid by a tyrosine radical engendered by the heme site with peroxidase activity, followed by the reaction of two oxygen molecules with the arachidonic acid radical, capitulating prostaglandin G2. Presently, cyclooxygenase 1, 2, and 3 are the three molecular isozymic form of cyclooxygenase enzyme are known. Numerous tissues articulate anecdotal echelon of COX-1 and COX-2, whereas COX-3 is supposed to be a splice variant of COX-1 with frameshift mutation, retaining intron 1 and favored with COX-1 variant, COX-1b or COX-1b. Both cyclooxygenase 1 and 2, also known as prostaglandin HS1 and prostaglandin HS2, act principally in the similar mode. However, selective inhibition can create a distinction in terms of side effects. In most of the mammalian cells, COX-1 is present as a constitutive enzyme, whereas COX-2 is an inducible enzyme, undetectable in the majority of normal tissues, but plentiful in activated macrophages and inflammation sites of the cells. The cyclooxygenase enzymes have recently been shown to be hypersecreted and upregulated in a variety of carcinomas and play a key role in carcinogenesis. Both cyclooxygenase 1 and cyclooxygenase 2 also oxygenate eicosapentaenoic acid and dihomo-gamma-linolenic acid to bestow the prostanoids of the series 1 and series 3, and these two are less inflammatory to that of series 2. Both eicosapentaenoic acid and dihomo-gamma-linolenic acid are competitive inhibitors with arachidonic acid for the cyclooxygenase pathways, explaining the fact why borage and fish oil as a dietary source of eicosapentaenoic acid and dihomo-gamma-linolenic acid reduce inflammation. In leukotriene biosynthesis, LOX5 catalyzes the conversion of arachidonic acid to leukotrienes via the incorporation of an oxygen atom at a particular place in the backbone of arachidonic acid. In leukocytes, especially basophils, eosinophils, mast cells, monocytes, and neutrophils, the lipoxygenase pathway operates during activation and resulting in the release of arachidonic acid from the phospholipids of the cell membrane by phospholipase A2, and finally transformed to 5-lipoxygenase by the lipoxygenase 5 activating proteins. The steps of the leukotriene biosynthesis include the conversion of arachidonic acid to 5-hydroxy icosahexatetranoic acid, 5-HPETE, by using LOX5 enzyme and LOX5 activating protein. In the next step, spontaneous reduction of 5-HPETE to 5-hydroxy icosahexatetranoic acid takes place. LOX5 acts yet again on 5-HETE to form leukotriene A4, which is an unstable epoxide. In the monocytes and neutrophils, the leukotriene A4 gets converted to the dihydroxy acid leukotriene LTB4 by LTA4 hydrolase. The LTB4 is a potent chemoattractant for neutrophils, performing at BLT1 and BLT2 receptors on the plasma membrane. In eosinophil and the mast cells, the LTA4 is linked with the tripeptide glutathione to form LTC4, the first cysteine leukotriene by the catalytic action of LTC4 synthase. Extracellularly, the LTC4, through the catalytic action of ubiquitous enzymes, forms LTD4 and LTE4. At the cell surface receptors like cis-LT1 and cis-LT2, the action of cysteine leukotrienes LTC4 and LTD4, LTE4, results in the contraction of vascular and bronchial muscle, increases blood vessels permeability, augments mucus secretion in the gut and airway, to employ leukocytes to the inflammation sites. Both cysteine leukotrienes and LTB4 are partially despoiled in neighboring tissues and eventually happen to be inactive metabolites in the liver. The biosynthesis of bile acid. Bile acid, also referred to as steroid acids, are primarily present in the mammalian bile, and when it is amalgamated with a cation, for example sodium, it is called bile salts. In other words, the protonated and deprotonated or ionized form are referred to as bile acid and bile salts respectively. In humans, taurocholic acid and glycocholic acid salts signify about 80% of the entire bile salts. The deoxycholic acid and cholic acid are the two major acids present in the bile. In the human intestine, bile acid, deoxycholic acid, and lithocholic acid, taurine, and glycine conjugates are present. The foremost purpose of the bile acid is to assist in the Michaelis formations that prop up dietary fats processing and an increase in the bile secretion exhibit the increased bile flow. In liver, bile acids are produced by the oxidation of cholesterol through cytochrome P450. They get stored in the gallbladder, water is removed, salts become concentrated in a conjugated form with glycine or taurine or with glucuronide or sulfate. In humans, cholesterol 7-alpha hydroxylase is the rate limiting step as the addition of a hydroxyl group at position 7 of the steroid nucleus takes place. During the intake of a meal, the gallbladder secretes its content into the intestine where bile acid emulsify the dietary fats. Other functions of the bile acid includes motivating the bile flow to remove liver catabolites, cholesterol elimination, emulsification of lipids, and fat-soluble vitamins to form Michaelis, which can be transported through the lacteal system and help in reducing the bacterial flora present in the biliary tract and small intestine. At intestinal pH, conjugated bile acids are more competent at emulsifying fats as they are more ionized as compared to the unconjugated bile acids. In most of these species, the biosynthesis of bile acid is a foremost route of cholesterol metabolism except humans. About 800 mg of cholesterol per day is produced by human body, and half of it is used for the synthesis of bile acid. In intestine, approximately 20 to 30 grams of bile acids are secreted daily, and almost 90% of it gets reabsorbed by the ileum through active transport and recycled by enterohepatic circulation. That budges the bile salt back to the liver and finally to the gallbladder. The process allocates a squat rate of daily synthesis but high secretion to the digestive system. The flat globules are broken down into tiny droplets by bile, and bile from the slaughtered animals is used in soap preparation. So, today in this lecture, we have learned about the biosynthesis of eicosanoids, including the prostanoids as well as leukotriene, how the prostaglandins synthesize, what are the different forms of prostaglandins and their role along with the catalyzing enzymes. Also, in this lecture, we understood the role of cyclooxygenases as well as lipoxygenases. In addition to the biosynthesis of fatty acid, especially the bile acid, as well as their esters have been